everybody, so I was having a little bit of trouble on the last video writing in the spaces, and I had started recording the next page, which is the giant chart, and it was a mess. So I'm going to do it a little bit different. You guys have the chart in front of you. I'm going to do it like this: proteins. So what is it that we need to know about proteins? First of all, the building block is that they are made up of amino acids. And the elements that you find in an amino acid is nitrogen, carbon, hydrogen, oxygen, and sometimes sulfur. So sulfur is a unique one. All right. They have very distinguishing features. First of all, when you look at a protein, you can notice NCC dash NCC dash NCC, and it keeps going like that. So let's talk about what this is. First of all, this is one amino acid, and the amino acid has an amino end right here. It has a central carbon that has a hydrogen, and then it has an R, which is the side chain. That's what makes each amino acid unique. There's 20 different ones. And then the carbon here on the end was a carboxyl group. So it's got a double bonded O. It lost the OH, and this nitrogen lost an H. So that formed water. So when you combine two amino acids together, it's dehydration synthesis. You dehydrate it, you remove water. And then this that forms between it, that's a peptide bond. So if I have a tripeptide like I have here, 1, 2, 3 amino acids, there's 2 peptide bonds holding it together. All right, next thing about proteins, you need to know their functions and examples. There are a lot of different proteins in your body. So some that you will think of right away: myosin and actin. They are in your muscles, and you think of those protein filaments, the myosin being the thick filaments and the actin being the thin filaments. Actin, there's a T in there. Okay, other proteins. Enzymes are proteins. Neurotransmitters are proteins. Hormones, not the sex hormones, but most hormones in your body are proteins. Antibodies are proteins. Channels are proteins. So there's a lot of different molecules in your body, and you can think of specific examples like insulin or acetylcholine or human growth hormone. Any of those kinds of things are going to be proteins in your body. All right, next thing I want to do, and again, I might not be following the exact same way the list shows it, but I'm going to go through all 4. I'm going to go through proteins, carbohydrates, lipids, and nucleic acids. So carbs. Carbs are made up of well, let's do the elements first, actually. C, H, O, and N. Oh, I know what it was, the building blocks. What are the building blocks of a carb? So the building blocks are simple sugars. And a sugar has, oh, goodness gracious, I don't want a nitrogen on there, sorry. C, H, and O. And they are in a 1:2:1 ratio. Now, as you have a larger carb, that 1:2, it might be off just a tiny little bit, but it's enough that you can tell, oh yeah, there's tons of oxygen in there, it's definitely a sugar. So let me show you what I mean by example. If we think of glucose, we used to draw glucose. There was, oh, sorry, here I am drawing the wrong thing again. Carbon, that was carbon 1. This is my oxygen. This is my carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6 is up here on a branch. So the formula for this would be C6H12O6. That's glucose. It's a monosaccharide. Now, if I want to put 2 of them together, what I have to do, there's the oxygen, I'm going to form a linkage right here, and it's called a glycosidic linkage. Again, I'm going to do dehydration synthesis to get them to pair together removing water. Now, when I go through and look at it, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, carbon 1, 2, 3, 4, 5, and 6. So I have combined all of the carbons. So I'm now at C12. And so one would think, well, this would be 24 and this would be O12. But it is not that. I have to remove H2O. I had to take out one water to connect them. So what I get is C12, H22, O11. So that's what I mean by the numbers might not work out as you grow larger than a monosaccharide. The numbers might not be a perfect 1 to 2 to 1, but it's far different from a fat where there's almost no oxygen. All right, what are the functions? So the functions, if you have a simple sugar, it is for immediate energy. And if you have a complex sugar, so like a polysaccharide, it is usually stored energy. Now I want to give you some examples. So let's do mono, di, polysaccharides. All right, monosaccharides, glucose, fructose, both of those are C6H12O6. They're just arranged a little different. Ribose and deoxyribose, those are both 5-carbon sugars. Disaccharide, lactose, is what you get when you put a glucose and a galactose together. Maltose is what you get when you put a glucose and a glucose. And sucrose is what you get when you put a glucose and a fructose together. And actually, I'm going to go down here. I'm going to make a new page. All right, I'm going to do the polysaccharides a little bit different. There are alpha glucose polymers and there's beta glucose polymers. The alpha glucose polymers we as humans like because we have the enzymes to digest them. So we can digest starch. We can produce glycogen. So this is made in plants and this is made in animals. They are both polymers, meaning repeating units over and over and over again. Alpha glucose, alpha tends to look like this with the O down, this with the O down, and these circles that I'm drawing are my glucose molecules. Beta glycogen or beta glucose polymers are things like chitin, which is what you find in an exoskeleton of a bug, or you can find it in the cell walls of a fungus. All right, so chitin has nitrogen in it, and that's what distinguishes it. And then the other one is cellulose. So we can't digest cellulose because we do not have enzymes that break down beta glucose polymers. So cellulose is typically what you find in a plant cell wall. All right, so we are going to move on from carbs and let's go to lipids. So when we're talking about lipids, we are talking about fats, steroids, waxes. All right, so the building blocks are usually fatty acids and glycerol. And I want to show you a little diagram of glycerol. Glycerol has 3 carbons. Every carbon has its own OH group. So glycerol is an alcohol. That's why it ends in OL. The other bonds are hydrogens. So that's the formula for glycerol. A fatty acid looks like this. C, 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 dot, 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 because there could be a whole bunch of them. C, C, C. And at the end, this is what makes it an acid. It has a carboxyl group on the end of it. So that's a fatty acid. All right, what elements? C's, H's, and O's. And I call it 1 to 2 to few. There's usually very little oxygens. And just think about how nonpolar a fat is. It's nonpolar because carbons and hydrogens tend to share their electrons pretty equally, and oxygen would share it unequally. It would pull on the electrons and make it polar, and fats aren't polar, so they really don't have many oxygens. All right, let's think about functions. What are fats for? They tend to insulate organs. They are a form of energy storage. They can make up the cell membranes and any membrane. So like the membrane of a mitochondria or a chloroplast or anything is going to be made of usually phospholipids. And what else? Sex hormones are steroids, so that's another function. All right, I know you're probably running out of space in all these boxes, but I'm going to do some diagrams. So some things that you really got to know. Phospholipids. So phospho tells me I got to have a phosphate group on it. So I'm going to start with my glycerol. 1, 2, 3. There's my OH. There's my OH. There's my OH. Now, in a phospholipid, instead of having 3 fatty acids form here, here, and here, not form, but attach in each of those places, what's going to happen is a phosphate group is going to attach right here. So O, P, O, O, O. Now, what can we say about this? This tends to be negative, which makes the molecule on this end be polar. So this is what we like to call the polar head. And then down here, I'm going to show those fatty acid tails. So first of all, I'm going to take an eraser. Sorry, I know you can't erase. I'm going to erase that line because the O is already bound. So it's going to be bound right there. So I got to erase all this. This would just be a hydrogen here. All right. What happens is it forms an ester linkage. So let me get a marker. Okay. If I have a fatty acid, this would be an O, this would be an OH. This would be carbon, carbon, dot, dot, dot. Same thing here. Carbon with a double bonded O, OH, carbon, carbon, dot, 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 dot. So what I want to do is take out water. So I'm going to take my eraser and I'm going to say, all right, water is one H, one O, and another H. One H, another H, and an O. If I remove the water, I can now connect these. That is called an ester linkage. And an ester linkage is when you have carbon to oxygen to carbon with a double bonded O and then the rest of a carbon chain. That is an ester. So my molecule has a polar head, and then look at this. These are going to be fatty acid tails. I just keep drawing carbons here. Now, this fatty acid tail, I have all single bonds. And if I have single bonds, it is a saturated fatty acid tail. Over here, I'm going to show a double bond. And a double bond is what forms the kinks in the tail. So this tail is unsaturated. All right, these tend to be very non-polar. There is a word for molecules that have both a, so here would be my whole molecule, 2 tails, 1 head. This side's polar, this side's non-polar. That's called amphipathic. Amphipathic is a molecule that has regions that are polar and regions that are non-polar. When they form a membrane, they form bilayers. So here's the layer on the outside. So I want that polar head facing out. And I keep going all the way around in a circle like this. These are probably how the first cells form. This is called a micelle. All right. But if I think about a big cell, it's not just a single layer. There's another layer on this side. So the tails face in and the heads would be here. I am not leaving myself much room for cytoplasm. You grab another color. The yellow in the middle here, this would be my cytoplasm. There's my cell membrane. Tiny little baby cell I drew, but you get the idea. Okay, what does a steroid look like? A steroid looks like 4 fused rings, and those make up things like estrogen, testosterone, progesterone. So those are steroids. 4 fused carbon rings tends to be very, very few oxygens. So you don't confuse it with a carb at all because it's missing all those O's. All right, and then the last thing that we need a picture of, I need a new color, triglyceride. All right, a triglyceride, tri means 3. So I have my glycerol, 3 carbons, OH, OH, OH. This time, 3 ester linkages. So here's my carbon with a double bonded O and an OH and carbon, carbon, carbon. And here is my carbon, double bonded O, OH. This is the carboxyl end of my fatty acid. Here's my fatty acid. And here's my 3rd fatty acid. Carboxyl end, carbon, 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 carbon. It keeps going. All right, I got to grab my eraser, and I got to remove water. So I'm taking out an H and another H and an O. An H, another H and an O. An H, another H and an O. So I've removed 3 waters, and now I connect these molecules. So all of those are my ester linkages. And what my overall molecule is going to look like is this. And the head isn't polar because it doesn't have a phosphate group attached to it. This is all a very non-polar molecule. And if the tails are literally straight, that is going to be a saturated fat in your diet. It is something that is a solid at room temperature like Crisco. It is not good for your arteries. It tends to build up plaque in your arteries and cause cardiovascular disease. Instead, something like olive oil is going to have a lot of kinks in the tails, which gives it a lot more flexibility and causes much less damage in the long run. All right, we are moving on to our last one, which is nucleic acids. Oops, AIC, acids. Okay, nucleic acids are what make up DNA, RNA, and ATP. They are all made of, so the building block is called a nucleotide. Sorry, go and put these in examples. I know it's down a little further, but your building block is a nucleotide. And a nucleotide has 3 parts. It has a phosphate group. So P, O, 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 O. The phosphate group is attached to a sugar. So this time it's going to be a 5-carbon sugar. There's the O. Okay. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5 would be right here on the branch. Okay, and then carbon 1 is attached to a nitrogenous base. Nitrogenous bases tend to look like this. And they usually have a bunch of double bonds, so these represent double bonds. They also have several nitrogens in the molecule, so it could look like that. If the nitrogenous base that you see here, oh, sorry, I'm hitting the, oh my gosh, I'm doing it again. Okay, hang on a second, bear with me. Get back to the screen I'm looking for. Okay, I'm sorry for that. I just know if I stop the video, it's going to take so long to get it back. Okay, so I have a phosphate group. I have a 5-carbon sugar, and I have a nitrogenous base. Nitrogenous base come in 2 forms. It could either be a double ring, or it can be a single ring. If it is a double ring, it is called a purine. Now, I like to remember pure is like angels and God. So, adenine and guanine, those are the 2 purines. Single rings are called pyrimidines. 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 They are just a single ring. So, these look like this. The 2 rings with the nitrogen. This is just a single ring with the nitrogen. T's, C's, and U's are pyrimidines. All right, and we already talked about examples, and they're going to be really important in terms of functioning for energy. Now, let's think of ATP, adenosine triphosphate. Adenine, we said, was a double ring, so I'm going to go ahead and draw a double ring here. Again, it's going to have a bunch of these, and it's going to have a bunch of nitrogens in here like that. Okay, that adenine is connected to a 5-carbon sugar. So there it is. And the 5-carbon sugar is attached to 3 phosphate groups. So there would be O's around it, and another P, another O, and another P, and another O. All right, so here's my phosphate groups like that. So there's 3 PO4s. So 1, 2, 3. Well, this bond right here and this bond right here are very high energy bonds. Break off one of them, and you are left with ADP. Oops, missed the D. ADP. Break off 2 of them, and you're left with AMP. So we talked about cyclic AMP, how it goes over and forms a circle. That's what happens. All right, so those are all of our macromolecules. Let's move on to the next one. All right, distinguish between hydrolysis and dehydration synthesis. So hydrolysis, hydro and lysis. That means that you are going to break apart the molecule, and in order to do that, you have to add water. So if I wanted to break that triglyceride into glycerol and 3 fatty acids, I would have to add 3 waters back into it to be able to break that molecule. Dehydration synthesis is when you are doing a building reaction. So in order to build the triglyceride, I had to remove 3 waters. So dehydration because you are removing water in the process. All right, next one. What is the role of an enzyme and how does it perform its function? So an enzyme is used to speed up a reaction rate. I want you to picture a reaction rate curve. It looks like this. We could start here and we got to go all the way up and then we end right here. So this is our reactants and here are our products. This energy that it takes to get to the top is called my activation energy. And what an enzyme does is it lowers the activation energy. So I'm going to use this yellow color. This could be my curve if I had an enzyme. I have to make sure I land at the same spot. I don't have to go up as high because it provides an alternate pathway for a reaction to occur. That alternate pathway usually involves the enzyme's active sites. So the active sites are what pull the substrate in and allow it to bind. How does pH, temperature, and concentration affect the function of an enzyme? First of all, pH ranges. There is always an optimal. When you go higher or lower than optimal, you tend to denature. And the reason why you denature is because if you have a really low pH, you're adding H pluses. If you have a really high pH, you're adding OH minuses. My protein is folded very specifically, and you'll remember one of the things about the folding is all about the charges. Well, the charges of hydrogen and the charges of OH might attract my molecule to fold in a different way. That's called denaturing. If it loses its overall conformational shape, it will not work anymore. All right, temperature. I'm going to erase, so I have room to do temperature here. Temperature also has an optimal, so I'm going to keep that there. If you go too high in temperature, you denature. You actually break IMFs. So the molecule starts to actually break apart and take on a new shape. If it is really cold, so you really have really low temperatures, it really just slows everything down so that there are fewer collisions, but it doesn't denature it. All right, and then concentration. So again, I'm going to erase. How does concentration affect how an enzyme works? So concentration. If you increase the concentration, you're going to have more molecules to collide with. So your reaction rate will go down. If you decrease concentration, fewer molecules means fewer collisions. That means a lower reaction rate. All right, design an experiment to test one of the factors from the last question on enzymatic activity. I am going to use H2O2 because that's hydrogen peroxide and it's what we used in our lab. And actually, I'm going to move forward to the next page so that I can go over the procedure from that lab so that I show how I can just alter one of these factors. So let's jump ahead. And it says Enzyme Catalysis Lab. The purpose was to be able to watch a reaction, time a reaction, and determine a reaction rate. All right, setup. First of all, we needed a substrate. Our substrate was H2O2, hydrogen peroxide. We needed an enzyme. Our enzyme was catalase. You can get catalase from any living tissue. Liver is used really often to get catalase from. We use potatoes in our lab. And we need an acid, and the acid is what we're going to add to stop our reaction because it's going to denature it. So what we did was that we first figured out a baseline, and the baseline told us how much H2O2 we had initially. Then we would set up timed reactions. So in all of these, I have H2O2. And what I do is I add the enzyme, I let it work for like 10 seconds. And after 10 seconds, I add the acid. That makes the reaction stop. Mix it up really well. Take out just a little bit of it, 5 milliliters of it, and do a titration. When you titrate 5 milliliters, you are determining how much H2O2 remains. If I know what I started with and I know what's remaining, I subtract and I figure out how much H2O2 was decomposed, how much was used. I do it all over again for 30 seconds and 60 seconds and 120 seconds and 180 seconds. And you can keep going and going and going. And what you're going to see is over time, H2O2 decomposed is going to start at pretty much nothing, and it goes up and up and up, and eventually it levels out. And the reason why it levels out, it ran out of substrate. If I wanted to get the reaction rate, I would take this section right here and I would calculate the change in Y over the change in X. So here would be my time in seconds. And I would determine, all right, how much H2O2, so this would be in milliliters. How much of this did I break down from this point to this point? And I would calculate my rate. Now, if I go back to this reaction, or this question, if I wanted to determine or design an experiment to test one of the factors, I use my same H2O2, I use my same catalase, I use the same acid, but this time I only run one trial. So pick one. I would pick a minute or maybe 2 minutes. And so I would do this, and you could have them with all different pHs. Test this one at a pH of 1, this one's at a pH of 2, 3, all the way up to 14, so you have a full range. And then again, you add your catalases, or your hydrogen peroxide's already in there, add your enzyme, let it work for 60 seconds, add the acid, stop the reaction, titrate it, figure out how much H2O2 is decomposed. Do the same thing for all of them, and then you can compare the effect of the acid. All right, next question. Explain how each form of inhibition regulates enzymatic reaction. Competitive inhibition, non-competitive inhibition. Okay, here's my enzyme. Okay, this enzyme actually has a substrate that fits right in the active site. All right, so there's my reaction. The substrate fits in. When it fits in, it breaks in half. So it's a digestive kind of enzyme, breaking that molecule. Now, over here, I have another molecule, and it looks a lot like that S, but I'm going to call it a CI. CI stands for competitive inhibitor. I could take this substrate and replace it with my competitive inhibitor. It fits in the same spot. So a competitive inhibitor is something that blocks the active site from the substrate. What's going to determine what I do more of? Whichever one of these I have more of. I have more substrates, I'm doing more of that reaction. I have more competitive inhibitors, I'm doing more of the blocking of the reaction. All right, a non-competitive inhibitor. So here's my substrate right here, And now I'm going to draw a new inhibitor. So here is my non-competitive inhibitor. And my non-competitive inhibitor does not fit in this same spot. Sorry, doesn't fit in here. Right? This does not fit here, but it does fit, and I'd have to turn it a little bit, but let's say it fits right here. When it fits right here, it causes a conformational change of the enzyme. The enzyme changes shape a little bit. Let's say it wraps around this a little bit. All right, when it changes shape, this active site is not available. That's how a non-competitive inhibitor works. And we study inhibitors a lot as scientists because they're the most common types of medicines. You want to block certain reactions and have them not happen in your body if they give a bad effect. So certain medicines that are taken are actually competitive or non-competitive blockers. Some of them are permanent, some of them are not. All right, allosteric inhibition. That is a word for any enzyme that has two forms. One form when it's active. So this might have a substrate and this might have an enhancer molecule. And when it has that enhancer molecule and that substrate, it is active and it is working. Enhancers could be anything from like vitamins to other proteins or other enzymes that interact with it. The other form of it is an inactive form. And the inactive form doesn't have the exact same shape. So it fluctuates back and forth between these two. When it's in the inactive form, the substrate cannot bind. All right, so an allosteric inhibitor is something that is, or an allosteric activator or inhibitor makes the enzyme either become more active or less active. All right, so that is the end of our biochemistry video. Make sure that you write down a list of questions that you have for me.